Let's talk about bacterium E. coli, a cornerstone of life science and biotechnology. Escherichia coli is a gram-negative, rod-shaped bacterium that naturally resides in the intestines of humans and animals. While some strains can cause disease, most are harmless and essential to biological research. It is the most studied organism on Earth, acting as a model system for genetics, molecular biology, and biotechnology. Discovered by Dr. Theodor Escherich in the year 1885, originally named bacterium coli, it was later renamed Escherichia coli in his honor. E. coli is simple and fast-growing, doubling every 20 minutes under optimal conditions. It is genetically well-characterized, with its entire genome sequenced, making it easy to study gene function. E. coli is also easy to genetically manipulate, accepting plasmids and foreign DNA readily. In industrial applications, it is scalable for mass production of proteins, enzymes, and drugs. In the medical field, E. coli produces insulin, growth hormone, interferons, vaccines, and more. Decoding the genetic code. Used in cell-free protein synthesis systems by Nirenberg and Mattei in 1961. This helped determine how RNA codons map to amino acids. Recombinant DNA technology. Herbert Boyer and Stanley Cohen used E. coli to express foreign genes in the 1970s. This led to the production of human insulin, known as Humulin, the first genetically engineered drug. Therapeutic protein production. Engineered E. coli produces human insulin, human growth hormone, interferons, and hepatitis B antigens. Humulin was approved by the FDA in 1982. Synthetic biology. E. coli is used to design biosensors, biofuels, and artificial gene circuits. It has been central in Craig Venter's work on synthetic genomes. CRISPR gene editing. Although CRISPR originated in other bacteria, E. coli is a primary testbed for editing techniques, plasmid delivery systems, and off-target analysis. Antibiotic resistance studies. E. coli is used to study how resistance evolves under stress. This research helps in the development of new antibiotics and resistance prevention strategies. Metabolic engineering. Modified E. coli is used to produce ethanol, butanol, and bioplastics. This supports sustainable biotech and green chemistry. Key experiment: DNA replication in E. coli. The Meselson-Stahl experiment in 1958 aimed to determine how DNA replicates, whether it is conservative, semi-conservative, or dispersive. Method overview. They grew E. coli in a heavy nitrogen medium, then transferred it to a light nitrogen medium and allowed it to replicate. They extracted DNA at time intervals and used density gradient centrifugation to analyze DNA weight. Findings. After one generation, the DNA was of intermediate density, with one heavy strand and one light strand. After 2 generations, the DNA was 50% light and 50% intermediate. Conclusion: DNA replicates semi-conservatively, meaning each new molecule has one old and one new strand. Why it matters. This is considered one of the most elegant experiments in biology and could not have been done without E. coli's rapid growth and manipulability. E. coli was the first organism in which many key components of DNA replication were discovered. Key discoveries: DNA polymerase, discovered by Kornberg in E. coli in 1956. Bidirectional replication with the origin at Oraceae, proceeding in 2 directions. Okazaki fragments, which proved lagging strand synthesis. Primase and helicase, first identified in E. coli. Replication fork visualized and studied in E. coli. Replisome, a multi-enzyme complex first seen in E. coli. Proofreading, with DNA polymerase III having 3 to 5 exonuclease activity. Significance. These discoveries laid the groundwork for understanding eukaryotic replication. They also inspired the tools used in PCR, sequencing, and gene editing. Expanded discoveries across life science fields. Genetics. The lac-operon discovered by Jacob and Monod provided a gene regulation model. Transposons, which are mobile DNA elements. Site-directed mutagenesis, enabling precision gene editing. Molecular biology. DNA polymerase, discovered by Kornberg, enabled in vitro DNA synthesis. Restriction enzymes such as Ecoli became gene editing tools. Plasmids, which are the backbone of molecular cloning. Biochemistry. Enzyme kinetics, studied through beta-galactosidase and tryptophan synthase. ATP synthase, with its mechanism studied in E. coli. Cell biology. The FTSZ protein, which forms the Z ring in bacterial cytokinesis. Replication machinery, including DNA polymerase III helicase primase and topoisomerase. Synthetic biology and systems biology. Toggle switches and gene circuits designed in E. coli. Minimal genome projects defining essential genes. Evolutionary biology. The long-term evolution experiment by Lenski from 1988 to the present, over 75,000 generations of real-time evolution. Nobel Prize discoveries using Ecoli. The Lac-Operon and gene regulation by Jacob and Monod, Nobel Prize in 1965. DNA polymerase by Arthur Kornberg, Nobel Prize in 1959. Restriction enzymes by Arber, Nathans and Smith, Nobel Prize in 1978. Genetic code cracking by Nirenberg and Khorana, Nobel Prize in 1968. Final summary, table Ecoli in modern life sciences. Field genetics with key discoveries like the Lac-Operon, transposons, and mutagenesis. Molecular biology, DNA polymerase, plasmids, and restriction enzymes. Biochemistry, enzyme regulation, and ATP synthase. Cell biology, cytokinesis with FTSZ and replisome components. Synthetic biology, toggle switches and artificial gene networks. Evolutionary biology, real-time adaptive evolution. Medical and pharmaceutical therapeutic protein production and vaccine antigens. E. coli is not just a bacterium. It's the workhorse of modern biology. The first step into the molecular world, and the foundation upon which countless breakthroughs have been built. Nature is ultimate.